The functions of a cell are determined by its DNA. Cells have to reproduce many times. In complex organisms, trillions of copies are made from one original cell. But when cells reproduce, they must replicate or copy their DNA. The structure of DNA reveals how trillions of copies of the DNA in one of your cells can be made, and be almost exactly the same each time. Francis Crick and James Watson discovered the structure of DNA in 1953. This breakthrough was on par with Newton's work in physics, but in our recent past. When Watson and Crick published the structure of DNA in a short article in 1953, they stated, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The fact that there are two DNA strands that are mirror images of one another suggested how copies could be made of each DNA sequence." Click here to see the letter Francis Crick wrote to his son about the structure of DNA. Each molecule of DNA is made of a template strand and a new strand. The template is used to make the new strand. The template strand is also known as the parent strand, since it came from the original DNA molecule. The new strand is also known as the daughter strand. Each strand has two ends, a 5' end and a 3' end. The two strands of DNA always run in opposite directions. They are said to be anti-parallel to each other. The template strands of the DNA molecule separate, and the new strands are made on the inside. The result of this process is two new DNA molecules, each having an old template strand and a new strand. This is called semiconservative because it conserves some of the old DNA in each copy. Click here to watch a video about DNA replication. Nucleotides can only be added to the negative OH end, the 3' end, and not the 5', so all new strands are made in the 5', 3' direction. Replication practice. What is the sequence for these new strands? Here are the sequences of the new strands.